Thank you, Kayla. Good morning, good afternoon, good evening. Thank you for joining today. We are honored to welcome Dr. Lebea, the Chief Scientific Officer at Tokayo Bio Ltd., to our GFRA webinar today. With over 17 years of distinguished experience in research and development, Dr. Lebea is recognized as one of Africa's leading scientists in the animal health sector. His extensive background covers both human and animal health, where he has excelled in molecular biology and biochemistry. As the lead scientific investigator and new business development contact at Tokayo Bio, Dr. Lebea has spearheaded innovative research initiatives and led diverse research teams driving advancements in infectious disease management. His contributions to the field are well documented with numerous publications in esteemed peer-reviewed journals. At Tokayo Bio, Dr. Lebea champions a One Health approach to infectious disease management, focusing on critical areas such as pathogen detection, biosecurity, biosurveillance, disaster management, and early warning systems. This vision is supported by two key groups, the Animal Health team and the Human Diagnostics group, both of which play a vital role in advancing public health and safety. Without further ado, Dr. Lebea, the floor is yours.
Okay, thank you very much. And thanks to GFRA for inviting me to share some of my thoughts in the program itself. I'm generally very excited to share my thoughts on the FMD field, which is very diverse and also sometimes can be very difficult. I'm generally more relaxed in two environments. It's one of them is not speaking in a webinar. It's more in the lab because I'm in control, but it's also in the field when I'm with the animals because they are always surprised to see me, like genuinely. So those are the two environments that I'm a lot more relaxed and happy to be working in. Now with foot and mouth, just to give a bit of background, in an African context, it's more of a burden in that the control and management of foot and mouth disease is always problematic. But it's also an opportunity in that we have an opportunity to lead in new and different ways within which foot and mouth disease can be controlled and managed, and also can be researched and new knowledge be known, especially with the mix of all the topotypes that one sees within the continent and the subdivisions of it. Next slide, please. If you look at value addition in livestock in the African context, I'll tell you as an African, in the continent, the number one employer is agriculture. Whether it's in plants or it's in livestock, Africans generally practice agriculture. Not always so successful, but generally we do that. Every time there's rain, people plow. Every time there's a little bit of forage, they rear and grow their livestock. I'll give this in the context of South Africa, where we have about 60-40 split between commercial farmers and small herd farmers. In this case, for the interest of everybody, commercial will be those people that own land and they have generally a high number of animals which they grow as a business, whereas the small herd farmers will be sometimes less than 20 cattle, and they grow them for subsistence or for semi-commercial reasons that now and then they sell. So it acts as a bank, literally. The status of many Africans is linked to the amount of animals or the type of animals they own. If you have a lot of cattle, you are wealthy. If you have some sheep, maybe you are, but not as wealthy as the one with a lot of cattle. If you have poultry, you are literally poor. You are trying to survive. That's all it is. Of course, it's completely different if you look at it on a commercial scale and how people live. Why is it like that? The pricing of the animals, what is it that you need to get as part of the cost? As much as one has to look at the disease profiles and what is it that afflict those animals. We have done some work. I've shown one of the publications there in Rwanda where we looked at risk factors for the incursion and spread of and persistence of foot and mouth disease. And here we were able to look at how far are the animals grazing, where are the watering holes, what happens during dry season, what happens during the wet season when everything is there. What about the interface between the wildlife and the domestic animals? And you'll find that the control and management of foot and mouth disease specifically in different countries varies a lot because the culture with which people look after their animals, especially cattle. For instance, in certain regions in Rwanda, the people look after those animals 24/7. In short, if you needed to know where the animals are, you just need to find where the owner is at any given time. Whereas I come back to South Africa, during winter, the animals literally mix, and it could be 2 or 3 weeks before the owner goes to find where the animals are. So to control disease, one has to also understand the context within the culture and the spirit with which the people look after their animals. What is it that they look after? What is it that they look at? And integrate that with whatever the scientific understanding and knowledge to try and look at that unique scenario within each country. Next slide, please. So if you look at foot and mouth disease specifically in the African continent, this may be a little bit outdated, but it's generally still true. You had mostly South Africa, Botswana, and Namibia that were exporting some of their cattle-derived commodities. And you had BVI in Botswana manufacturing FMD vaccines, and another company in Morocco that also was manufacturing vaccines for foot and mouth disease. I think there's about 6 or 7 different types of combinations in general within the African continent of foot and mouth disease topotypes. And therefore, the amount of vaccines available to deal with specific foot and mouth disease viruses is not always adequate. The surveillance before there is an outbreak is also varied depending on the regulatory authorities within that country. And most of the times, once the vaccine is bought, it's not always adequate in enough quantities or boosted in the right time, depending on what the matching of those viruses to the vaccine might be. And generally, there's no post-vaccine monitoring once the vaccination itself has been completed. Hardly do we do any stamp out in Africa for obvious reasons. That for most of the small herd farmers, which is the bulk of the people within the continent, the compensation is not possible, and it becomes a lot more complicated. But if you look at the number of animals within the continent, and you look at the amount of funds that could be generated if there was a better control of notifiable and reportable diseases, that could contribute a lot more of foreign direct income by exporting some of those commodities that are linked to the livestock, and would improve a lot of the socioeconomic issues in the rural areas where the animals generally grow, and not necessarily in the cities. And if you look at the depressed nature of the villages and the rural areas where the animals are, there is a big gap that needs to be filled, and application of technology to make sure that the socioeconomic status of such people can be improved, not necessarily by growing more animals, but by controlling and managing disease, and foot and mouth diseases being one of those. Next slide, please. So I had worked for the CSIR, which is the Council for Scientific and Industrial Research in South Africa. It's a parastatal of the South African government, which specializes in research and development to the benefit of the South African population. And while I was there with some of my colleagues, we had developed an isothermal PCR. In this case, it included the instrument itself that we use for that, which is battery operated, and it can be linked to the mains. And we have since been able to work with different companies, and we have lyophilized reagents, which makes it easier to not require refrigeration all the time. The instrument is portable. We have been able to develop as well an IT system that goes with it, which we can run as an app on the phone, and we're able to run the instrument from there and have the data itself put within a database. And then you can also integrate the instrument. You can run it as a single instrument. It's about 16 samples. Or you can run up to 5 instruments together on a laptop, and they would run up to 80 samples as if it was one machine that is running. So you can run a lot more samples in one go, or you can run a few. Now, it depends. If you run it in the lab and you have a lot of samples, you want to run a few of the instruments as one instrument. But if you're in the field where you want to know a yes, no, maybe, you want to run one instrument at a time. We have been able to use this primarily in Rwanda. We have run a few of the curated samples in South Africa. We have now a pilot program that will be running in one of our provinces with a mobile DSL-3 lab that will assist us in being able to set up where the outbreak is, and we can run these instruments at will and be able to manage the disease. How it works, obviously, it's as shown on the bottom left there. It's more of a secular and repetitive amplification of the genome sequence that you have used, because isothermal PCR uses a set of 4 primers, as in 2 pairs. It is a lot more specific, although it is about 1 log less sensitive than your PCR is. But for the sake of detection in the field where you have an outbreak, it's about 2 logs more sensitive than you require in any case. So there are no issues with the sensitivity or the specificity of the instrument. And with the application and the telephonic link to the instrument, it becomes very easy to have communication linked between the people in the field and the analysts in the lab, and in most instances, then the regulatory authority that actually is responsible for reporting such a disease to the OIE. On the right, you can see there what an isothermal PCR profile between 3 different serotypes, and the profile in the middle, and on the left side where you see a peak, that would be a second derivative of the graph in the middle, so that you can pinpoint the quantities of the virus that you are busy measuring. Next slide, please. This is just the example of the mobile app that we use linked to the instrument. You're able to log in your name and your password, and we're able to see the program itself, and we can download to look at the full graphs like I showed you in the previous slide. But what it helps is that it also can pinpoint the actual GPS location where you took the sample and where you ran it. But you also have a communication portal within the app that you are able to communicate with people in the field, similar to what you do on a WhatsApp. If you needed more pictures or you needed another sample to be taken, say for sequencing, or a clinician that we work with wanted to see certain symptoms or profiles that will help them to make diagnosis, the system is designed in such a way that it is able to do all that. Next slide, please. Now, what are some of the developments that we've done? We've looked at lateral flow. So we look at it this way. If you are to control foot and mouth disease specifically with an understanding to export or to send some of those commodities outside your country, it means you have to maintain a green zone, as in free of disease without vaccination, preferably. Sometimes it can be free of disease with vaccination, depending on the countries that you need to send to. But it means there has to be constant surveillance. And to do surveillance, there are different types of ELISAs. We specifically work with IDVET, and we are able to use their ELISAs, which are very good for FMB. And we have developed our own isothermal PCR. This is specifically if you have a suspicion that there may be an active virus circulating. Then you want to have the virus and literally be able to amplify. So we call that outbreak containment. And if you did find a positive animal at a specific area, you want to take the same instrument and look within the area, say, depending on the understanding that I've showed you in the beginning, where the watering holes, how far do they graze, what is that area, which is the area they're likely to go. So you can put boundaries based on the understanding of the grazing patterns of the animals in the area that you are. And that will allow you to know, if it were to spread, which are the areas that you could be looking at. Then you can take your same instrument and work from the outside going in according to the spatial understanding and the grazing patterns of your animals. Depending on whether the outbreak is there or not, there could be vaccination that is required. And generally there we would work with other companies. Recently we have initiated some discussions with BVI in Botswana. Not yet formalized, but we are looking at how else do we work together to make sure that we combine both the surveillance capacity and the vaccination campaigns. We obviously within the country work with Honest Export Bioproducts, that's OBP, which has other vaccines outside foot and mouth disease, because to look at the general health of a herd, it's not only FMD you must look at, you must also look at other diseases that otherwise might affect the health of your animals, but also can affect the exportability of that herd. Then we participate in vaccination campaigns, but we also have developed some lateral flows, which we don't use for the diagnosis, but rather for post-vaccination monitoring. The instrument on the bottom left as well is part of the lateral flow strip reader, which then gets the picture and pixels around the band that is on the strip, and those get digitized, and they're also sent back to the database the same way that you would have done for isothermal PCR. We have developed a mobile lab. It looks a bit similar to the one on the top left, but that's not exactly ours. Ours will be launching officially in November this year, which then would be used mostly within the areas and the provinces that we know would need to export from there. Next slide, please. We do have as well, besides for the mobile unit, container labs as the ones that you can see on the top right of the picture. And in that one, we're able to put it in a big truck and take it as a main lab in the area that we know we need to do control of disease. The three doors, as you can see them, one is more sample reception, and the two doors that are furthest from each other, one is the middle one would be the extraction room, and the furthest one would be your PCR room. But in the inside, they are linked by a hash that means the people working inside do not necessarily need to walk in and out to be able to transfer the samples from each one. It is negative air pressure control, and so the containment is quite valid within that. And we find that for the purposes of having an impact in the area where you have problems, it is easier to take either a containerized lab or with your mobile lab as well, so that you set up where the disease is rather than go and fetch samples and bring them to the cities, which may be very far from where the problems are. At the bottom there was one of the study that was done, but this was in poultry, but we think it might be applicable at some point in foot and mouth disease as well, where using the surveillance tools that we have, it means you are able to get that virus and you can characterize it by sequencing. And should it be that specific genes have been identified that you can put into a vector that already is known or has been used as a carrier to use for vaccination, you can then grow that within your bioreactors and make a preliminary vaccine that, depending on the type of disease, and not necessarily foot and mouth disease in this case, you might have what we call an emergency vaccine. This was one of the PhD studies that Dr. Marema has done. That's the lady on the left there. And in poultry, this was actually very useful, and it means from detection to early formulation of that product could take something like 35 to 40 days. And we think these are some of the strategies that in future might be worthwhile adopting to try and curb the outbreak of such diseases, including FMD, very early on, rather than running very expensive vaccination campaigns each time, even where people might not necessarily have the funding to do so. Next slide, please. We do as well, actually on the left there, that is the inside of the container lab that I had shown you. In the middle and the right, we also, given that we move labs to the areas that might not be suitable for laboratories, it means there's also a risk of testing the disease, and for real, you find it is FMD, but again, be spreading it because you do not have the correct containment, the correct disinfection, or the fumigation. So the more virus you bring into one place and you don't do the proper way of cleaning up, you might inadvertently be spreading the disease yourself rather than be assisting. So for that, we've run 2 or 3 programs, one with ozone, where we're shown, especially during COVID, how do you use the ozone within the FMD or virally concentrated lab to fumigate. We've also used another product, escape kill disinfectants, from one of my colleagues, Bruce, and with the combination of the two, where we do the fumigation almost every day after the end of the session, it helps in us knowing that within the lab itself, how much is the risk for the virus being still available after the work, and how do we clean those labs, how do we change up, and how do we go in and out, because it's just as critical and serious as the detection itself. So while the mobility helps with the agency with which one runs to the field, it is just as critical to make sure that the environment in the area where one is operating does not become a contaminant itself. Next slide, please. There is also a program that we've put together. This is between the University of Cape Town, the CSIR, my former employer, ourselves, and to a certain extent, the IDVET as well, and other programs where we are looking at within South Africa, but also within the SADC and Africa in general. How do we share some of the knowledge that we have to make sure that it is applicable and usable within other countries and other regions within the continent? I'll give you a simple example. If I run to another country, even within South Africa as well, the understanding of whether you're able to get a sample or not depends on the language, the culture, and how you talk to those people. Sometimes somebody says, yes, I'll bring the animals, but the cultural understanding is that he will not bring them. Unless you grew up there and you understand what they're saying in a way that you have grown up in it, you can lose valuable information. So it is important that as one works, in Rwanda, we work with the Rwanda Agriculture Board. They've been very helpful when we needed to sample buffaloes. They would get a squadron that came with us and help us with that. GFRA in Zambia, Raina Njinga Mbande University in Angola. These are some of the colleagues that you have to work with to make sure that within the group of people you work with, they have an understanding. But a quick one here is that if you look at your left and there's like a green background, that's what we generally do. This is surveillance and sampling, real-time disease detection. This is now with both the lateral flow and the isothermal PCR, and virus identification through sequencing and other means that we would have in our labs. But when it comes to viral typing and small-scale production of the prototype vaccines that may be made based on the viruses that one picks in the field, that we look to other colleagues that are more specialized in that to help us, but we have our own input and what else can be done or not. This includes Sagita Biotech in Belgium, who have their own vector and what they look at based on the measles, and how else do you make it compatible for bovine as opposed to humans. There are other companies, I've spoken of OBP and BVI, that if that had to go large scale, then we have collaborations with them to say, are you able to manufacture it in this way or that way? We obviously participate in vaccination campaign, in disease monitoring, and also in assisting with compiling the OIE reports. That is obviously not our job as a company, but for the regulatory authority within that country. But the bottom line is, how do you get FMD and other viral vectors out of the animals through control, through vaccination, through surveillance, and through development of other detection techniques that allows you to be functional and be fully automated, even in the area where you might not have a laboratory. Next one, please. it sounds like I'm done. I just want to thank GFRA for inviting me here. The DSI, which is our Ministry of Science and Innovation, Technology Innovation Agency, for some of the funding, the CSIR for the development of the technologies, and EXIN, which is our partner in Australia, in the hardware of the instruments that we use in this program. Thank you.
Thank you, Dr. Lebea, for your insightful presentation on the advancements of surveillance for foot-and-mouth disease, vaccine development, and all the collaborative engagements with other countries in Africa. I'm going to start with one question. Perhaps you mentioned this before, but what are the main challenges that are contributing to the inadequate ability of FMD vaccines in Africa?
Well, one of the issues that you get, especially within the continent, is that there's generally scarcity of vaccines, as in the money itself is not available. And if the funds are available, the vaccine that is being used is not necessarily matched to the outbreak that is there within that country. And sometimes very well-meaning countries in the developed world would donate vaccines that are completely irrelevant for that area. And by the time you go back for the next outbreak, you find topotypes that you don't even know where they come from, because you have animals that are vaccinated with a vaccine that does not necessarily assist. And then the outbreak goes through like wildfire. So until there are no animals to infect anymore, that's when it stops. And that makes the control and management a lot more difficult, based on the lack of resources and sometimes acceptance of just any vaccine that is available rather than the one that is matched to the outbreak.
Thank you. Melanie?
Thank you, Giselle. Good day, everyone. And thank you, Dr. Lebea, for that very interesting presentation. Perhaps we could just elaborate a bit more on the FMD management. You touched on it now. What do you think should then be the focus on FMD management within the African context? What is your opinion on that?
I think, personally, I think if the focus was on surveillance, in other words, what is out there in the field, and people know exactly which strains are there, and even their drift is understood, it would be very easy to know when there's an outbreak, which vaccines from where one should be getting, compared to every time it's more or less we're reacting to an outbreak rather than having the knowledge of what is available within that area, so that our management process is seamless. So in most areas, you'll find only when there's a confirmed outbreak, then we start running, where do we order vaccine from? How much should it be? How many doses? So half the time, we are chasing the disease rather than being ahead of it, so that we're able to deal with it adequately. And while one can be tempted to say that's a function of the availability of resources, it's not, because it's cheaper to do surveillance than to buy vaccines every time to be able to manage the disease. And half the time, we would know plus minus which outbreak is likely to happen based on where the FMD is endemic and what are the strains that are available in that area.
So you feel that there's not adequate surveillance being currently done, in your opinion, in different countries in Africa?
Yes, it is done almost as a program rather than as a surveillance to understand the response that one must do in an event of an outbreak. So you may find, say, a country like ours, where maybe over 1,000 tests have been done a year. But to what effect? Why are we testing those? But if I say we have a red zone towards the Kruger National Park, what are the strains that we have? So if we had an outbreak Is it similar to those strains? And if it was, depending on where the outbreak is, how many vaccines do we need? Generally, that answer is not there. It becomes a matter of how much funding do we have to buy how many doses, and then now we can run around and try and do that. So I think the surveillance with an understanding to guide the management of the control and management of the disease would be a lot more helpful. And that is not just for South Africa, it's throughout the continent.
Yeah, thank you. Giselle?
Thank you. We have a question from Mariano Perez. Are there any efforts going on around epidemiology studies of susceptible wildlife species?
Is this as in the continent, or is just a general question?
I think it's a general question.
Look, for instance, when we first did foot and mouth surveillance in Rwanda with the team there, one of the things we did was to try and see within the buffaloes what was circulating, if any. And then we had to look at within the outbreaks that were there, what type of strains were found. And then for any other animals that one is able to find, you are able to take a sample, or depending on whether it's a susceptible animal or not. But now and then there are programs that I've seen. I've seen the ARC with some programs in the Kruger National Park with buffaloes as well. We've done some in Rwanda, but generally they are far and few between such studies. Although I think they would be critical, especially where you suspect wildlife and domestic animal interface.
Thank you, Melanie.
Thank you. The next question is from Anne Kellett. The question is, how important do you see public-private partnerships for FMD management in Africa? Will this also assist with potential funding through commercial farmers to improve surveillance?
Thanks for that. I think it's probably the best way to go forward. Why do I say that? If I grew up in a village up in the north in South Africa. I still visit my parents there. I know that half the time the people there will think twice before paying for an animal that is sick, because they need to have the money to take their child to a clinic. So it is not sustainable in such an area, especially if you look at the subsistence farmers. Why am I answering it using subsistence farmers while we're talking about commercial? Because the disease doesn't know any boundary between commercial and subsistence farmers. And so the disease, FMD, is likely to circulate a lot more uncontrolled within small herd farmers compared to the commercial farmers. So the best way to control it would require that within those subsistence farmers, there's even more rigorous management of the disease, while the commercial farmers are able to pay for that and make it as part of the cost. So that partnership must include increasing the ability of the rural poor to be able to manage their animals in such a way that they're exportable. Whether they're exportable by selling to the commercial farmers that then export, or they export directly themselves, that's neither here nor there. But once you have a conduit for those animals to be exported, which means better funding or better money for the owner of the animal, you are now getting to a point where surveillance and animal treatment becomes sustainable financially, because then there is a source of funding that comes through that foreign direct income of the export. But if it's just a partnership for the sake of it, then you're again sitting with the same problem where there isn't enough funding to procure the tests, to get the guys to go to the field, or to even buy vaccine for that matter.
Thank you. Giselle?
Yes, we have a question from Toby Tuthill. When resources are limited, presumably control of FMD is not always a top-level priority. Given the transboundary nature of disease, how do neighboring countries manage different prioritization of disease control?
Yeah, that's a difficult one. I'll give you an example with Lesotho, not Lesotho, as in a non-export country, and its boundaries being totally sharing with South Africa. And as contrast with Botswana and Namibia, which share borders with South Africa, but with other countries as well. What do we see there? When before we had this current big outbreak of FMD in South Africa, it means farmers interchange animals. When they need more, they buy in Botswana. When Botswana needs more, can buy in South Africa, Namibia, and vice versa. Therefore, while the three countries are running their budgets differently as far as disease control is concerned, they are mutually benefiting from each other by keeping each other's animals clean. Now, once you have South Africa with an FMD outbreak, you can literally see the risk that it puts to Botswana or to Namibia, the restriction of the animals' movements and all that. So I think once the governments and the ministries understand the action between the disease control of theirs, the budgets they're putting, and of their neighboring countries, it becomes easier. There was once in Botswana, not in Botswana, in Lesotho, an outbreak of anthrax. And we were still doing our work in Lesotho as well. One of the things we did was literally to talk to OBP and to talk to the Animal Health Directorate and explain this. And you could see that immediately looking at the risk of what that could do, the Ministry of Agriculture was able to talk to OBP, and I remember about 20 or 30,000 doses were released for free as a donation to the neighboring country to help with curbing or controlling the disease. There were some testings that were done there, and there were exchange of personnel as well to assist with that. So for as long as the outbreak of disease does not have a negative impact besides for the animal health itself, then there'll always be problems. But once the animal health is linked to the sustainability and financial benefit of the country and the communities that are there, there's a very good chance that that cooperation will happen. In lots of countries, you'll find that, I know, especially going into Central and Eastern Africa, almost every year there's an outbreak of FMD. It doesn't bother a lot of people. It is now normal. But why is it normal? Because as long as it continues like that, but there's no fear of reduction in the finances, especially as far as foreign income is concerned, then there's very little action that is prescribed to that.
Thank you. I have a follow-up. Are there any specific educational initiatives that can be implemented to perhaps raise the awareness about FMD and the impact that it has on livestock?
Yes, and yes, it's 2 yeses. The first one is, I think I showed in my presentation as well that luckily within the South African context, we've been speaking to different organizations, and we've been also speaking to other governments and public institutions, including universities within SADC region, where we are saying, why not set up some sort of a center of excellence, not necessarily to go after FMD, but to say within the One Health program, FMD included and others, how do we deal with FMD as an example in different regions? Again, based on culture, based on the way that things are done, but also based on what do we need to develop technically. It cannot be one solution fits all. What are the type of kits that you can use in certain areas? I can give you an example in one country that I won't mention, where it is so hot in summer, and they go and fetch the vaccine, and they put it in the van, and they drive for 5-6 hours with ice packs. By the time they vaccinate the animals on the 3rd day, that vaccine literally doesn't work. Now, how do we go about it? How do we develop a vaccine that they would reconstitute where they are? How do we make sure that the cold chain, besides for getting fridges and freezers and take everywhere as it is, the electricity is unstable? How do you work around that? So I know IDVET, through the consortium we have, wanted to help with the farmer education, and also with the CSIR and UCT and other organizations. We are looking at the Center of Excellence to also put younger minds within the program to develop things that would be usable within the different scenarios, other than just getting the samples from the field and bring them to the city for analysis and run through that. So there is hope if you look at the type of private-public partnerships that are being put together. Also, the outreach programs and trying to understand the farming methods from the people that practice it, rather than just impose what we think works in other countries, especially in developed countries.
Thank you. Melanie?
Thank you. We have a question from David Lazarus. What is your advice to the policymakers on the impact of spillover of FMD from previously diseased, endemic, or controlled areas to disease-free areas within a country?
We have struggled with that problem for the longest time. Sometimes it's not just the spillover. Governments or the public sector is rightfully so looking at specific programs. In other words, you cannot just change the policy in one day. You have to go back to say, if we have a scenario A or B, what is it that we do? In our case, what we have done, and understanding as an example that the control of notifiable disease falls squarely on the Ministry of Agriculture and not any private companies running around or any institutes that do not have the mandate. But what we do as well is we have put at least one postdoc that works on looking at what are the policies from other countries that have better control of this. What are the policies that we have in our own country? If we were to have a policy in an area that is a problem, let's say spillovers where there might not be anything, what should it look like? What should we be considering? Because it is easy to blame public servants, certainly in my country, but how are you helping? How are you assisting to make sure that the containment or the control of disease works? So we have to get to a point where we say to our public counterparts, here is what we think a policy would look like. And here's our thinking, here's ABCD, but you draft and you study it according to what is it that generally the government will look at. And I think that is becoming more and more an area that as scientists we have to look at and say, how can we assist rather than wait for a policy to be put in front of us and we apply it.
Thank you. Giselle?
Thank you. The next question is by Abdi Mahamoud Elmi. Are there any cases of foot-and-mouth disease in the camel family that have been recorded in Africa or elsewhere?
That's the easy one for me. I don't know it. I haven't worked on it. So it would be interesting to see if there's such, but I haven't heard of one and I haven't worked on it.
Thank you. Melanie?
Okay, we have a question from Salah Zahi. Don't you think that any vaccination strategy cannot succeed in controlling FMD in the absence of a reliable identification and traceability system, especially in the context of our African continent, where cross-border, transhumane, and extensive livestock farming are predominant in many African countries?
It's a, I almost want to say it's a yes and no, but it's mostly a yes. In that, when one says traceability, again, is what do we mean? Do we mean a tag on the ear? Do we mean a sequence of that animal so that even if it's dead, we can identify that piece of meat back to this is person A's cow, so it shouldn't have crossed? Or is it talking? This is what we find, okay? If you say to a group of people, and here, because I work more in the rural areas, if you say to a group of villagers within specific villages, if somebody brings animals from outside, let me know. If somebody has a sick animal and it's reported, let me know. If there's ABCD, let's work on it. They understand that very well. But if there's nothing in it for them, it doesn't make sense, because why am I running around informing this guy every time there's ABCD for what? But now superimpose that scenario to where you have cleared an area into a green zone. In other words, into an area where vaccination is not required anymore. And by policy, it's identified as an area that is FMD free, and therefore they can export. That could be a difference of another 50 to 75% more money per animal. Now, all of a sudden, introduction of an animal in that area means I'm losing my livelihood. Somebody's animal sick is threatening my livelihood. I'm not reporting so that I'm seen to be reporting. I'm reporting because I'm preventing my animals from getting sick. It is traceability, because every person that has those animals know their animals. And if there's a new animal that doesn't belong to them, they can quickly report it because they want to check before it makes their animal sick. So when we say traceability, and we look at our situation within the areas that we live, we must also identify, what do we mean? Are we talking some satellite chip in the ear that we're able to see it's this or that? Are we talking methods and ways that we're able to identify each animal according to the needs and arrangements in that village? So an animal or a bull is not going to walk 120 kilometers in a straight line in 3 months. No, it rotates and walks around the same area. Everybody either knows whose it is or the owner knows what it is. So I think when it comes to traceability, the answer is yes, we must have that. But secondly, we must develop strategies that do not necessarily need us to pay an arm and a leg just to know, to have that sophisticated type of traceability, which might not work in reality. We have to also think of the livelihood of those people that have the animals, to say, we want traceability for what? What is it that they benefit if they enforce that traceability? In the villages, I find that I will not even know. Somebody will call me and say, so-and-so's son-in-law has brought 20 cows from somewhere where they shouldn't be here. Then already you know. That is traceability. You go and check them. You don't fight the people, but you see, are they sick? Are they not? Whatever. That is traceability system.
Thank you. Giselle?
Thank you. I think we can squeeze one more question. The question is, has any successful risk-based vaccination strategy been implemented in an African country, given the limited resources for purchasing vaccines? What are the best methodologies for developing such strategies?
Look, in the continent, personally, I think if one only looks at animal health as the health of the animal, then that strategy fails. But if one looks at animal health as linked to what I said initially in my earlier slides, as for us living in the villages, that is our bank account. So if now I have animals that I must now take my money and look after them, it just doesn't work. But if I look at it as in our collective looking after our own animals helps in the amount of money I can have when I sell my animals, then you have a winning strategy. So risk-based would have then to go with, when do we need that vaccine? Which vaccine is it? Because we obviously cannot do the stamping out like they would do in Europe. Why? Because if you do that, then you must compensate for the animals that you have culled. And generally, there's just no money to do that. So I would want to say, with a strategy that emphasizes a lot more on the surveillance itself, whether it's passive or active. Passive here, I'm talking about for so many animals in such an area, so many animals, according to the statistical understanding, must be sampled for us to know if there's FMD or not. Or active is, during this time with the water scarcity, these animals walk far, and these are the main watering holes. So the area within which the animals interact is a bit bigger. Then you know how to expand your surveillance to look and control it much earlier, including if we are sitting in South Africa and in Lesotho, there's an outbreak. It's a matter of time before it's in the Free State province. What do you do within that area in terms of the vaccination and the surveillance that you need to do? So the best strategy, according to me, is still to be proven, is you have to do as much surveillance as you can. Your policy must be congruent to your reaction strategy in the field. Your preparedness for such an outbreak in every region must be informed by what you see on the ground, including your sentinels and the people that react on the ground early on, and how do you fortify that area to make sure that the disease, as one of the questions has come through, doesn't spill over without necessarily understanding what is happening on the ground. In that way, you limit the amounts of vaccine that has to be bought and how far you can go with the control itself.
Thank you, Dr. Lebea. I think we have reached the end of this webinar. We thank you for your participation, and thank you everybody for participating. We still have a few questions left, but we will ask you to answer those questions, and then the answers will be posted in the GFRA website. Melanie?
Thank you so much, Dr. Lebea. We really appreciate you presenting to us today. Really interesting, very nice discussions as well. We're going to hand over to Kayla now to round up for us. Thank you.
Thank you so much. I want to say thank you to Dr. Lebea for joining us today and presenting for this webinar. Thank you to Melanie and Giselle for your work in helping to organize and facilitate the webinar. And thank you to everyone who joined. We really appreciate you taking the time out of your day to support the GFRA and our webinar series. As you leave the webinar, you'll see a link come up. Short questionnaire. I'm going to ask that you all take just a few more minutes to fill out that questionnaire. We really do use that to continue to improve this series, especially looking at 2025. We'll absolutely be using your feedback to shape the future of the GFRA webinar series. Thank you all.
Thank you. Bye.
Thank you. Bye.